Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Dear students of 12th class, subject Botany, Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, our today's topic is the basic features of Mendelian experiments and techniques applied. Dear students, first of all, we will study the basic features of Mendelian experiments. Dear students, you know that the Gregor John Mendel is considered as the father of genetics, and he made various experiments on the edible garden pea, and now the basic features of his experiments are: number one, the plant selected by Mendel for experimentation was called the Pisum sativum, or in general English we can call it the edible garden pea. The plants were found to differ in certain easily detectable traits. These traits, known as contrasting traits, always appear in two opposing conditions, one dominant and other recessive. Character means a heritable feature, while as each variant for a character is called a trait. For example, if the size of the plant is considered, the size is the character and tall or dwarfness is the trait. During experimentations or during the experiments on Pisum sativum, Mendel noted 7 pairs of contrasting traits in pea plant. So dear students, you should remember and you should get to understand that the plant selected by Mendel was the Pisum sativum, and the plants were found to differ in certain easily detectable traits, and those traits are called the contrasting traits, and they appear always in two opposite conditions, one dominant and other is called the recessive. Character here means a heritable feature, while as each variant for a character is called a trait. For example, size is character, tall or dwarf is trait. And Mendel noted 7 pairs of contrasting traits in pea plant. Now we will show these 7 pairs of contrasting traits in a tabulated form, and on the top of the table we can write contrasting traits noted by Mendel in 7 characters of garden pea. Contrasting traits noted by Mendel in 7 characters of garden pea. The first column of the table is the serial number, and second is the character. Third one is the dominant traits, while as fourth row is called the recessive traits. The serial number 1 is the height, which is the character, and if it is dominant then and is tall, then it is shown in abbreviated form capital T, and while as if it is dwarf, that is it is a recessive trait, then it is can be dwarf and is shown by small t. In the same way, the second character is the position of flower, where the position means where the flower is located. If it is axillary, then it is shown by capital A, but when it is terminal, it can be shown by small a. In the same way, the third character is the color of unripe pod. If it is green, then shown by capital G, and if the recessive trait is yellow, then it is shown by small g. The fourth character is the form of unripe pod. If the dominant trait is inflated and is shown by capital I, and the recessive trait is constricted and is shown by small I. The fifth character is the color of seed coat or flower. Dominant trait is colored and is shown by capital C. Recessive trait we can write in recessive trait column, we can write this white, that is the small c. The sixth character is the color of cotyledons. Dominant trait is yellow, shown by capital Y. Recessive trait is green and is shown by small y. The seventh character is the form of seed. The form of seed character, dominant trait is round and is shown by capital R, and the recessive trait column we can show the wrinkled, that is the small r. Dear students, these are the 7 contrasting characters which have been noted by Mendel. These contrasting traits, which are 7 in number, are found in the pea plant. Now, the second basic feature of its experiment is the plant being bisexual and shows self-pollination, so the traits were preserved generation after generation. Over the generations, it shows considerable genetic uniformity. This is the second basic feature of Mendelian experiments, that the plant being bisexual and shows self-pollination, so the traits were preserved generation after generation. Over the generations, it shows considerable genetic uniformity. Dear students, the third basic feature is that the Mendel easily cross-pollinated the flowers by removing the immature stamens and dusted the pistil of this flower with the pollen of desired plant. It means that Mendel cross-pollinated the flowers by removing the immature stamens from them and dusted the pistil of this flower with this pollen of desired plant. The fourth basic feature is the plant is annual herb with short life cycle and can be cultivated easily. The plant produces many seeds in one generation, which helped in drawing correct conclusion. So after these basic features of Mendelian experiments, and by detecting the 7 contrasting characters or traits from the garden pea, then the Mendel applied the experimental techniques. Those experimental techniques which have been applied by the Mendel on garden pea, we will discuss in our next lecture tomorrow, inshallah. So till tomorrow, stay home, stay safe. Thank you very much.